Preparing for tomorrow's workshop at Amsterdam University Medical Center at the Immunology and Molecular Biology department. Let me show you what are the things that I've done. Let me just collect my thoughts. Okay, first thing first. I created, with using Vicena, all the instruments that there are in their core facilities. I asked it to go and look at all the instruments, confirm that they are there with the proper links to the instruments. So, for example, microscopy, cell and in vivo imaging, single cell and genomics, biochemistry, data analysis softwares, and all the things that they already have. I created a list here. I asked Vicena to go and do research, and it created this list for me. So this is important because we want to do research with the tools that is already there. When experiment designs and all of that gonna be done with the instruments that are there. The next thing that I did, now invert this in this list into functionality list. What can be done? What type of experiments can be done using these instruments? Go and verify each of the specs of these instruments from the vendor's website and what can be measured. For example, if it's antibody or for word scattering, and all of the low-level experiments which can be done using this equipment. Of course, this is not an exhaustive list, but this is essentially most of the experiments that can be done using the setup that is out there, right? And all of them verified. When you ask to design an experiment, I can create essentially what are the steps, how to do those. Then I thought, what is the next step? They get the data. How do we analyze that data? How do we get that data and get signals out of it? So what I asked Vicena to go and install those packages for those instruments to do the data analysis, what data analysis tools they are. Go and find the open source versions with the Python SDK or any of that that we can essentially use and upload the data. And this is the notebook that is created. For example, cell type analysis and also mass spec analysis and all of that. Of course, here was first we started with the synthetic data, like fluorescent microscopy, synthetic data. But then I asked, go and give me the data sets, real data sets, so I can use it to show with real data sets. And here is with the real data sets that I downloaded that real data set. Here it is, PBMC 3K raw, which is essentially a raw data set. And here is this marker gene based on real data set and the visualization based on real data set. And this is what you can see is being doing while I'm talking to you. So this way we can follow the whole workflow, and I can show you the whole workflow in a biology lab, in an immunology lab. So I have now ways to do literature review. I can design experiments based on the lab instruments that I have, the functionality map, and I can analyze the data, get the insights, get the information using Vicena in an agentic way. So I can follow the whole process. And tomorrow I'm going to show this at the Amsterdam Medical Center. Wish me luck. You know, every time that you try to show a demo, a lot of things can go wrong. And I'm prepared for that. And I'm doing all my work So I know exactly what I'm gonna show and how I'm gonna show it to you. Let me see. I might record that and put it out there for you. Thank you very much. Have a nice day.